Mr. Chairman, ladies and gentlemen, it's a great pleasure for me to be here with such distinguished faculty. I'm an interventional cardiologist in West London, and my main interest is actually the discovery of biomarkers which underlie premature coronary artery disease, with a specific interest in biomarkers amongst Indian Asians. So, when one thinks about Egypt, we are drawn to these amazing human endeavors, ingenuity, creativity of the pyramids. But clearly, ancient Egypt offered much more. Homer, in his Odyssey, remarked that in Egypt, the men are more skilled in medicine than any of humankind. So, the contribution of Egypt, ancient Egypt, to medicine is enormous. It is the first formally recognized ancient civilization to practice medicine in a formal and well-documented manner. It also paved the way for Imhotep, an Egyptian priest who is widely regarded as the father of medicine. But you know, Magdi would have given a good run to Imhotep for his money. So Magdi Yaqub established heart transplantation in the UK and became the world's leading transplant surgeon. Established and became the master of the Ross procedure, pioneered the modern arterial switch, promoted the use of LVADs for transplantation, established the Heart Sciences Center, the Institute of Cardiac Research, the Chain of Hope Charity, humanitarian surgery across Africa. Amazing intellect, contribution to clinical research, recognized by the Royal Society. And when I came to the airport yesterday, I think even Imhotep could not have achieved this, which is have his photo in the baggage hall of the airport. So, as I said, the LOLIPOP study aims to identify the environmental and genetic factors that contribute to incident cardiovascular disease in Asians. Why is it a problem? It's a problem because 60% of the global cardiovascular disease burden by 2020 will be in the Indian subcontinent. Currently, Indian Asians have a twofold excess risk of cardiovascular disease compared to Europeans. The situation with diabetes, 50% of the global diabetes burden by 2030 will be in India, and currently Indian Asians have a 3- to 4-fold excess risk of diabetes compared to Europeans. India has been subject to vast invasions over the years, starting with Cyrus the Great, Alexander the Great, but none have inflicted as much damage on India as these two conditions here, cardiovascular disease and diabetes, which each account for over 2 million deaths in India every year. Not only is this a big burden, it's a big burden amongst young people. Earlier cardiovascular deaths, 50% in India, 25% in the world. It's estimated that by 2030, the loss of productive working life will be tenfold higher in India compared to the US. And people talk about the advancement of India and about the economic resurgence of India, but these conditions will account for an estimated 1% minimal reduction in GDP. So the projected foregone national income to heart disease in India, as you can see, is around $200 billion a year. So why is this the case? Why are Indian Asians at increased risk of cardiovascular disease and diabetes compared to Europeans? The first study, which went into quite a lot of detail with this question, were the studies in West London conducted by McKeague and his team. And these were observational studies in around 1,500 Indian Asians, 1,500 Europeans, and he discovered that the common risk factors, smoking, blood pressure, and cholesterol, were not consistently elevated amongst Indian Asians, in the various subgroups of Indian Asians, compared to Europeans. Yet they all had an increased risk of cardiovascular disease. Oops, I think I've... Which is the point? Okay. So all the major risk factors pointing in different directions, yet all had increased risk. And he concluded that because the major risk factors were not consistently elevated in the direction of risk, these did not account for the excess cardiovascular disease in this population. Instead, diabetes and related metabolic disturbances were consistently raised in this population compared to Europeans. And using regression analysis, he concluded that diabetes and related metabolic disturbances account for 70% of the excess risk of heart disease in this population. Enter Salim Youssef in 2004 with the InterHeart study in about 15,000 cases and 15,000 controls worldwide. From this population, he had about 2,000 South Asians, and he found that the 9 common risk factors, smoking, dyslipidemia, blood pressure, diabetes, obesity, and so on, all increased, were all risk factors for myocardial infarction in South Asians as in every other population, and collectively they accounted for over 90% of myocardial infarction. So his message was quite clear. It wasn't a new message. This message had been available earlier in other populations, but it was an important message because in Indian Asians he challenged the view that diabetes and insulin resistance were accounting for the majority of the excess risk. The limitation with the InterHeart study was that it was a case control study. It was not a prospective design, and it did not look at incident events, and it could not address the differences of the reasons why there was excess risk in Asians compared to Europeans. So the SABRE study in West London was a prospective effort, 16-year follow-up of Indian Asians and Europeans, and in this study, the SABRE investigators documented a 2-fold increased risk. 2-fold increased risk in Asians compared to Europeans. And this 2-fold risk could not be accounted for by either conventional risk factors, hypertension, diabetes, cholesterol, nor indeed by the metabolic disturbances. So, in actual fact, in this population, contrary to the findings of McKeague, the reasons underlying the excess risk for cardiovascular disease and diabetes remain unknown. So, it was with this in mind that we set about to identify the reasons underlying the excess risk of diabetes and heart disease in this population. Now, our study is based in West London. In West London, the Nile does not run very deep, and in West London, the pyramids don't always turn out well. But we do have some interesting historic monuments, but we also have in West London a population of around 500,000 Indian Asians who are exposed to this increased risk, who have been the subject of our study. So our study was based on a collaboration. We had no money when we started. We collaborated with everyone we could, the community, the hospital, the trust, the industry. We employed nurses. And with a team of 52 GPs, we spread our nurses across all practices. And each nurse went to each practice to see 5 patients per day in each practice, and we collected this data. The LOLIPOP study is now over 30,000. We collected data on past medical history, family history, alcohol, physical activity. We measured their blood pressure, height, weight, waist-hip ratios, did the ECGs. We collected their blood sample. We did their biochemistry. So some of our team, seen around this slide. Specifically, I want to draw attention to John Chambers, who's hiding behind this clock, who is a co-investigator of this study, who has worked with me for over 20 years and works with Elio in his department. And at the top, Paul Elliott, also with James Scott. So this study has generated over 2 million blood samples, which we archive in a warehouse which we had to buy because we were thrown out of the storage space in the hospital. And we've currently got in excess of several billion data points in this study. One of the things that we've done in this study is actually when we've examined the patients, they've been kind enough to participate in this study, we have given feedback to our patients concerning their risk factors. It's very unusual for the patients in London to get feedback when they participate in research studies. So our patients have had dedicated, tailored reports landing on their doorstep within 5 to 7 days of having participated in the study. It helps to bring them back 10 years later when we want to follow them up. So what have we found? Well, we've found an excess risk of heart disease in this population. We've found an excess risk of diabetes in this population. 2-fold increased risk of heart disease and a 3-fold to 4-fold increased risk of diabetes. I just want to share with you some of the slides. So in this slide here, the prevalence of coronary heart disease with age, the Europeans here in the empty symbols, the Indians in the filled symbols, and you can see that the prevalence of coronary heart disease is at least 2-fold higher in Asians compared to Europeans. But this twofold increased risk of CHD is not just present in people who are 60. It's present at every single age group. It's present in women and it's present in men. And when we look for adjustments of the reasons why this might be the case using multivariate regression models, we find that when you correct for age, classic risk factors, physical inactivity, obesity, and the metabolic syndrome, there remains a 2-fold increased risk in this population. What of diabetes? The story is very similar. The prevalence of diabetes, Asians at the top, Europeans at the bottom. At every single age group, there's a 3 to 4-fold increased risk of diabetes in Asians compared to Europeans, in men and in women. And when you correct and adjust for BMI, waist-hip ratio, physical inactivity, HOMA, you find that there remains a 4-fold excess risk of diabetes compared to Europeans. So this was worrying. So we decided to change our strategy. We decided to adopt rather agnostic systems biology type approach, and we said we're going to subject our population to whole genome sequencing, scanning, proteomic scanning, metabolome-wide scans, and to see whether we can discover the markers which might predict the increased risk in this population. So at that stage, a number of loci had been discovered for heart disease, and we asked the question, is the excess risk of heart disease in Indian Asians explained by genetic variation? So the known common variants associated with coronary heart disease, we asked ourselves the question, is the increased risk of heart disease in Indian Asians accounted for by the presence of shared variants, which may have a higher allele frequency and may have a greater effect size? This slide summarizes what we found. So this is a population that we studied. This is a forest plot. It shows the association of the SNPs with coronary heart disease in Indian Asians. The direction of effect of the SNPs with coronary heart disease in Indian Asians was similar to that in Europeans. We looked here at the allele frequencies and the effect sizes. Allele frequency here, effect size over here. Indian Asians, odds ratios. Indian Asians on the horizontal axis, Europeans on the vertical axis. Horizontal axis, Europeans on the vertical axis. And the diagonal line, you can see that there was no difference, really, between the allele frequencies we found in Asians compared to Europeans. And if anything, the effect size of the SNPs was greater in Europeans than in Indian Asians. So known common genetic variants did not appear to account for the excess risk in this population. So the emergence of the Human Genome Project, the International HapMap, and the related research tools of high-throughput genotyping and bioinformatics has led us to move into genome-wide association studies. And genome-wide association studies, very simple. Essentially, it uses the LD structure of the HapMap. It selects SNPs to tag and differentiate haplotypes. Here you see at locus 1, 3 haplotypes. They're differentiated really by the purple and the blue SNPs. And in locus 2, the 2 haplotypes differentiated really by either this or that SNP. So these SNPs get genotyped in cases and controls. You look at the frequency of each of these in the 2 populations, and you replicate to see where the signals might lie. And so we did a genome-wide association scan. We did a genome-wide association scan to discover Indian Asian specific SNPs, and we combined our work with European genome-wide association scans and investigated several thousands of individuals. As you can see from the slide, we replicated in over 40,000 individuals, and we found novel loci accounting for heart disease. But we did not find that these were specific to Indian Asians. So we found Indian Asian SNPs, we found European SNPs. The SNPs that we found in Indian Asians were also present in Europeans. They were not specific to Indian Asians, and they did not account for the excess risk of heart disease in this population. I show this slide because we then moved on briefly to look for reasons underlying sudden death. This was a collaborative effort with Magdi and his team, Cesare here. And this picture is taken from our research office, which hasn't got an enormous amount of room, so when we all get together, there's hardly any room. But it keeps us going because it's a cozy environment and it's a good academic exchange. So in this slide, we investigated the genetic factors underlying cardiac conduction. We identified SCIN10A to be the locus associated with prolonged PR interval. We found that SCIN10A was expressed in human heart, which we obtained from Harefield through Magdi. We demonstrated that this variant was associated with defects in conduction, principally prolonged PR intervals, and we demonstrated also that it was associated with heart block and ventricular fibrillation. We did not demonstrate that this was present in excess in Indian Asians compared to Europeans. So, now in the interest of time, I'm not going to go into diabetes, but the similar story holds. Associated SNPs with diabetes, allele frequencies and effect sizes very similar. We do not find reasons why known variants might explain the excess risk of diabetes in this population. Similarly, we did a genome-wide association scan in diabetes, and here we discovered 6 new loci for type 2 diabetes, and these 6 loci had the same frequencies, same effect sizes in Asians compared to Europeans, and despite scanning the genome, we did not find reasons why diabetes was present in excess in Asians compared to Europeans. And so we have gone on and looked at a whole other facets risk factors which might account for it. We've discovered a lot of stuff, but we have not discovered the reasons underlying the excess risk of type 2 diabetes and heart disease in this population. So this bit here, shared variants accounting for heart disease and diabetes, now disproven. So we said to ourselves, is it possible that there are actually Indian Asian specific variants? In other words, variants which we may not find in Europeans. So we've gone ahead to do whole genome sequencing. So one of the reasons that the current efforts have not discovered variants underlying diabetes and heart disease is because these arrays are based on European variants. The scans are based on European variants, and the completeness with which these variants capture genetic variation in other populations is not known. So on this slide here, you see what's known as a multi-dimensional scaling plot. It's a way of clustering people who are genetically similar. And you can see that clustering here are Europeans, Africans, and Chinese. So we use principal components, it's a statistical methodology to look for genetic variation which is able to distinguish, differentiate regional and ethnic backgrounds, and we plot our data here amongst Indian Asians compared to that of the HapMap data, and you can see that Indian Asians lie in a completely different genetic space compared to Europeans and Africans and Chinese. So this gives rise to the thought that there may well be separate variants which underlie health and disease in this population. And indeed, this finding here, which is a discovery of a 25 base pair deletion in a cardiac myocyte binding protein present only in Indian Asians, not present in Europeans, accounts for a 7-fold increased risk of heart failure. So spurred on by this kind of discovery, we've gone ahead to do whole genome sequencing in this population. And the idea here is to systematically go through the whole genome to identify every single variant. And we have here discovered over 500,000 new SNPs, of which over 2,000 are coding SNPs which lead to changes in proteins. And the idea here now is for us to take away these SNPs, put them into a separate array, and go through the whole series of experiments again in cases and controls to see what genetic factors might underlie risk in this population. We have also extended our work to looking into the epigenetic basis of coronary heart disease and diabetes and the metabolomic basis of coronary heart disease and diabetes. And time does not allow us to go into this data. But our overall strategy is to make discoveries here and to integrate all these discoveries together and to then move on into the follow-up of the LOLIPOP study. This is now coming up to a 7.5-year average follow-up, over 180,000 person years of follow-up, a significant number of cardiovascular events. The study is very well powered to discover risk factors which are specific to this population. And of course, the predictive tools which are necessary. So what's the point of all this? The point is that we want to understand the disease mechanisms which underlie heart disease and diabetes, which ultimately might help discover drugs and also predictive tools. So this study here, we applied the Framingham function to looking at reported and observed risk in Indian, Asian and Europeans. We found in West London the Framingham reported and observed risks were very similar, but when we applied the Framingham function to Europeans, as did others, we found a big gap. So these functions and these risk factors were not allowing us to predict the reasons of underlying increased risk in this population. So the idea is that hopefully with some discovery, we might be able to bridge this gap. So in the meantime, the problem rages on, and we have to find an interim solution. The interim solution in India and Indian subcontinent largely has been driven by REVASC and REVASC and REVASC. The Prime Minister went through his second or third bypass operation and became a celebrated story in India. They make bigger and bigger and bigger stents in India. But I was very taken by this slide from Capel and colleagues, which showed the deaths averted from 1980 to 2000 in the US population, and it shows that some of the risk factors have got worse, may have therefore increased risk, but the principal reduction in risk looks as if it's actually being driven by management of risk factors and to a lesser extent through treatments and specifically much less through REVASC. So with this symposium in mind, maybe for these vulnerable populations where we do not have all the answers at present, maybe in these populations we've got to attack vulnerable plaques which may not progress onto heart attacks in your face, but maybe look towards stability, maybe towards management of LDL levels and risk factors to such rigorous levels that you get them into the regression range. So I hope that I have given you an overview of the LOLIPOP study and where it's going with its discovery strategy in an attempt to look and discover novel biomarkers. I believe that this kind of strategy is suited to the population in Egypt because the phenotype of the Egyptian cardiovascular disease resembles that of Indian Asians as it does in the Middle East. It's a problem which is looming and is going to grow in this country, and it would be very good for
your time is up
to move on with a prospective population like LOLIPOP. So finally, I'd just like to acknowledge the collaborators. Thank you very much for listening.